Okay, so what you have studied in muscle tissue? I have seen few of the students, you have written in your practical notebook also. Few students, they have written in the practical notebook. So what you have studied about the muscle tissue? Because see, the epithelial tissue, the connective tissue, whatever you have studied, okay, the concept of the muscle tissue is completely different. It's not same. That's why I'm asking what you have studied, because based on that we'll start the revision. Okay, have you seen the slides of muscle tissue? For example, skeletal muscle, cardiac muscle, smooth muscle. Have you seen the slides? This time talking about this. Not yet, right? Okay, don't worry. Then today we'll see those slides also along with the slides of trachea. Okay, and from now onwards every day in histo class 2 tissues will be taken simultaneously. Okay, or else I can't complete the syllabus. So if you see about the muscle tissue, so these are the contractile tissue of the body derived from mesoderm. Okay, so these are contractile tissue, mesodermal in origin, and it creates about 40% of the weight of the body. Okay, next if you see the muscle tissue, actually it is a fiber. The muscle tissue, they are mainly made up of fibers, muscle fibers. Okay, now together so muscle tissue consists of fibers of muscle cells connected together in sheets and fibers. So together these sheets and fibers known as muscle, and they control the movement of an organism and having contractile function. So different types of movements whatever are taking place. Okay, movements doesn't mean that only the locomotion whatever you are doing, other movements are also there. Okay, so that is also governed by the muscles which are present in the body. Next, what are the characteristics of the muscle? As a whole, if you see all the muscle fibers, what are the characteristics of the muscle fiber? First of all, irritability is there. That means ability to respond to a stimulation. Whenever you are giving some stimulus, it will respond to that stimulation. That is the irritability. Then contractility, that means after giving a sufficient stimulus or after getting a sufficient stimulus, it will contract. Okay, that is the contractility. Then extensibility means ability to stretch. That means it can be lengthened beyond the resting length. Then elasticity, ability to return to the resting length after being stretched. So these are some important properties of the muscle fibers. Okay. Now before going to the microscopic structure of the muscle fiber, you should know some terminologies which are related to the muscle fiber. Okay, basic terminologies are there. First of all, you should know that the muscle fiber, so as I said, if you take any muscle, first of all they are made up of muscle fibers. If you take one single fiber, it is nothing but a cell, and that is called as myocyte. Okay, one single fiber, it is a cell that is called as myocyte, and that particular cell is elongated in one particular direction. That's why they are termed as fiber also. But actually what is that? That is nothing but a cell, and that is called as myocyte. Okay, next, inside that cell an important protein is there. Can you see the name of the protein? That is the myosin protein, which helps in the contraction of the muscle fiber. Okay, next, plasma. So if a cell is there, cell will be having plasma membrane, right? In case of muscle fiber, it is called as sarcolemma. Next, cytoplasm is called as sarcoplasm. Then ER is called as sarcoplasmic reticulum, and mitochondria is called as sarcosome. Okay, so these are the terminologies, because now onwards I will be using those terminology. Okay, so please don't get confused. Same only, though they are present in muscle cell, that's why the name is different. Okay, now each muscle fiber again is covered by connective tissue. Okay, so connective tissue coverings are also present in the muscle fiber. Next, these are some functions of the muscle as a whole. Okay, so mobility is there, very important. Whatever movements you are doing, starting from the locomotion, starting from whenever you are chewing something, when it goes into the intestine, the peristalsis, everything is done by the muscles only. Okay, so stability, heat regulation, circulation, digestion, respiration, vision, maintaining the posture. Okay, these are some important functions of the muscle. So I am talking about as a whole. Okay, not about one particular muscle, as a whole. Next, you know that 3 different types of muscles are there in our body, right? What are they? Skeletal muscle is there, cardiac muscle, and smooth muscle. These are the three different types of muscle fibers present in our body. Now already you know the examples also. Can you give some examples of skeletal muscle? Yes, skeletal muscle, whatever muscles you have studied in upper limb, lower limb, those are the examples of skeletal muscles. Okay. Smooth muscles, what do you think, where they are situated? Yes, in the internal organs and in the heart. Okay. Now, next what we have to study? Next we have to study about the individual muscle fiber. Okay. So under microscope, whenever you will see any individual muscle fiber, how you will identify, what are the important identifying features, how you will differentiate them, okay, that you have to know. Okay, so what do you think, what is the important identifying features for skeletal muscle? Yes, striations are there. Next. Okay. Okay, not nucleus, nuclei. Then striations, cylindrical, oh, fibers. Okay, what else? Almost, not exactly. And another important feature which you have not mentioned, they are not branching. The individual muscle fibers, they are not branching. Okay. So already you know the important identifying features for skeletal muscle. Okay. That whatever you have said, these are correct. Okay. And that you can see under normal microscope. Under 40x also all those things can be seen. Now beyond that, what you have to know? Any idea? Beyond that, what you have to know? Beyond that, you have to know about the striations, how those striations are formed. Yeah, actin and myosin is the smallest unit. Between that, what are the things present? That, Yes. Okay. So I hope you have heard about dark bands, light bands, or also called A band, I band. Okay. So that you have to know. Okay. And then gradually we'll go to the filaments. Okay, the myofilaments. So if you take one muscle fiber, inside that myofibrils are there, multiple myofibrils. If you take one myofibril, it is made up of myofilaments. If you take one myofilament, that is made up of alternating actin and myosin filament, or also called as thick and thin filament. And due to the arrangement, alternate arrangement of this actin and myosin filaments, okay, it appears striated. Okay, so like this you have to go in. So first one muscle fiber, then myofibril, myofilament, and then those actin and myosin filaments. Okay, so skeletal muscles are also called striated. Why? Because of the striations, and they are also called as voluntary muscle. Already you know that skeletal attachments are there and is responsible for locomotion. These muscles are also called voluntary because they are under control of the somatic nervous system. Okay, so see, I was talking about this. So this is individual muscle fiber. This is an individual muscle fiber. Okay. Now inside the muscle fiber, can you see the fibrils are there? Myofibrils are there. Now if you take one myofibril, then this is the appearance. Again inside that you can see the myofilaments. Okay, where you will be getting the actin and myosin. So this is how you have to study. So up to this much muscle fiber and the cross striation, that can be seen under compound microscope. In 10x also somewhat you can see. In 40x you can see the striations easily. Beyond that you need electron microscope. Okay, in order to see the myofibril, and after that in order to see the myofilaments. Okay, electron microscope is required. Let me show you the, have you seen such kind of diagram? So this is the diagram showing the skeletal muscle fiber. Okay, skeletal muscle fiber. H&E staining of skeletal muscle fiber, where you can see the muscle fiber. Can you see, this is one muscle fiber, this is one muscle fiber. So they are not branching. Okay, you can't say that always they are parallel, almost they are parallel to each other, but they are not branching, just like the cardiac muscle. Next, you can see the striations. Can you see the striations? Dark and light striations alternately. So these are present, and this can be seen. Next, if you see the nucleus, it is multinucleated, and towards the periphery you can see the nucleus. Okay, they are situated towards the periphery. That much you can see. Okay, so basically during the time of exam, I am talking about practical exam, this is the most important thing which you have to know. that first whenever you will see the slide, you have to identify the cross striations. Okay, along with that you have to identify the nucleus, where the nuclei are situated mostly at the periphery, and the individual fibers, they are not branching. Okay, that much you should able to identify under the microscope. Okay, now the most difficult part in the skeletal muscle is the striations, the most difficult part. Okay, so as you can see, it is made up of long cylindrical fiber. Already you have mentioned this. The length and diameter of the fiber varies greatly. The length approximately highest it becomes 30 centimeter, and the diameter is 10 to 60 micrometer. Okay, next you can see the nuclei are elongated, okay, and lie at the periphery. Now the cytoplasm, also called as sarcoplasm, filled with longitudinal fibrils, and these are called as myofibrils, which I have shown you. That means we are discussing about one individual muscle fiber. Okay, we have to take one individual muscle fiber. So inside that, what are present? Myofibrils are present. Now in the transverse section, the myofibrils often appear to be arranged in groups, and these are called as field of Connective. Okay, so these myofibrils, if you take a TS, they are appearing in a group. Okay, and this is called as fields of Connective. Now the myofibrils are uniformly distributed throughout the fiber, and the most striking features are the presence of this transverse striations. Okay, which I have shown you just now. Now after staining with H&E, you can see alternate dark and light bands, and they are also called as A band and I band. Okay, now can anyone tell me what is the concept behind this A band and I band? Very good. What is the meaning of isotropic? Okay, that means you know the concept. Everyone, you know the concept, right? So I am not discussing it again in the class. So that is depending on the isotropic and anisotropic nature. Okay, for that you need polarized light, not normal light. Next, some other portions are also there. Okay, that is alternate dark band and light bands are also there. Now in between those dark band and light bands, some other bands are also there. For example, if you prepare the slide properly, then what will happen? Further details can be seen. Running across the middle of the I band, there is a dark thin line, and that is called Z band. So middle of the I band, okay, again there is a dark line or dark band, that is called as Z band. So one Z band, another Z band. In between that, the area which is there, that is called as sarcomere. It is called as sarcomere. Next, at the middle of the A band, traversed by a lighter band, that is called as H band. Actually, it is not a band, it is a zone, H zone. Okay, and again at the center of the H zone, another band is there, that is called as the M band. Okay, so these are nothing but the arrangement of the actin and myosin filaments. Okay, this appearance are there. Now they run transversely across the whole muscle fiber because of the corresponding adjoining myofibrils. Okay. Next, as I said, the myofibrils situated between two consecutive Z bands are called as sarcomere. Okay. Here you can see, so this is A band, or also called as dark band. This is light band. Again dark band, and again this is light band. Now this is dark band. At the center you can see H zone is there, and again at the center of the H zone you can see M band is there. Now here at the center of the I band, and another zone is, another band is there, that is called the Z line. So between this Z line and this Z line, the area which is there, that is called as sarcomere. Okay, now see, every time I am showing you that, so these are the muscle fibers. Okay, this is the skeletal muscle fiber. Now inside that you can see these are the myofibril. Now you have taken one myofibril. Okay, inside the myofibril, can you see these cut sections? What are these? These are called as myofilaments. Okay, so like this we are going to the smallest part, smallest unit. That is the smallest unit. First, this the entire part is the muscle fiber. Inside that you can see the myofibrils are there. You have taken out one myofibril where you can see those alternate dark band, light band, sarcomere, all those things. Okay. Next, if you see the cut section, you can see numerous myofilaments are present. Okay. Now, what do you think? What will be present in the myofilament? Just now I have mentioned thick and thin filaments, or actin and myosin filaments. Now for that, what you need? You need ultrastructure. Ultrastructure means electron microscope structure. Okay, so the electron microscope structure of myofibrils. Okay, so these are the structural and functional subunit of the muscle fiber. Now arranged longitudinally in the muscle fiber and run throughout the length of the muscle fiber. Now these myofibrils consist of bundles of myofilaments. Okay, and these myofilament consists thick or also called as myosin and thin filament also called as actin. Okay, so like this the myofilaments are formed. Now here you can see this is how the arrangement of the actin and myosin filament. Okay, so these are the thin filament, these are the thick filament. Okay, so you see alternately they are placed, and because of that only those dark band, light bands appears, and then you can see on normal microscopy the striations. Okay, so this is the basic of the striations. Next, you know that two types of fibers are there, type 1 and type 2, and another type is that it is called intermediate type. Do you know that in skeletal muscle the fibers which are there, okay, types are there depending on how they are contracting. Okay, fast is there, fast twitch fibers are there, slow twitch and intermediate. Okay, now the appearance of those fibers are also different. Now for example, if it is type 1, they are also called as slow fibers. Okay, now they are red fibers. Why? Because large amount of myoglobin is present in the mitochondria. Okay, that's why the color is red in nature. Next, they are slow twitch fibers because they contract slowly. They are resistant to fatigue, means easily they will not get fatigue. Now can you give some examples? Examples of the postural muscles of the back. Now for example, you are sitting, okay, you can sit for at least 2 hours, 2.5 hours, 3 hours. Just imagine whenever you are watching movies, at that time you sit at least for 2 and more than 2.5 hours or sometimes 3 hours while watching the movie. You don't get tired after watching movie. Okay, at that time also your postural muscles are working, it's maintaining the posture. Okay, how it can maintain? Because they are type 1 or slow fibers, because the contraction is very slow, steadily. Okay. Next, some other fibers are there, type 2 fibers. They are white fibers. Okay, they are thick fibers, contain less amount of myoglobin in the mitochondria, but high amount of glycogen is there because they have to contract very fast. Now can anyone give me some examples of these type 2 fibers? Examples of type 2 fibers. Fibers of the extraocular muscles of the eye. Okay, extraocular muscles of the eyeball. Okay, then fingers or the digits muscles of the hand. Okay, just imagine whenever you are playing this mobile game. Okay, continuously you can't play for 1 hour. Within half an hour you will feel that paining. You have to take rest. Okay. So I am giving the examples which you can apply in your day-to-day life so that you can understand. So these are the type 2 or fast fibers, and in between them intermediate fibers are there. Okay, so can anyone give me the example of those intermediate fibers? Mostly they are the fibers of lower limb. Okay, so they are in intermediate. Now here you can see the comparison between the red fibers and the white fibers. Just go through it, you will understand. Okay, the comparison between the red fibers and the white fibers. Along with that, the examples are also given. Now, as I mentioned earlier, that the connective tissue covering is there for skeletal muscle. Okay, now you can see 3 layers of connective tissue covering for the skeletal muscle. Can you see over here? First you can see the epimysium is there. Then you can see the perimysium, and then you can see the endomysium. This is the endomysium. Okay. Now what exactly is this connective tissue covering? Any idea? Have you heard about fasciculus or fasciculi? What is this? If you take any skeletal muscle, it is having a fascicular arrangement. What is the meaning of that? Yes. So if you take the skeletal muscle, generally they are arranged in bundles. Okay, so many muscle fibers together they will form bundles, and those bundles together will form a particular skeletal muscle. Muscle fibers are there, so many muscle fiber will form one bundle. Like this, so many bundles will be there, and those bundle will together form a skeletal muscle. Okay, this is the normal arrangement of skeletal muscle. Now, each and every step, the connective tissue covering is there. So if you take entire muscle, which are made up of those collection of the bundles, that is also covered by a connective tissue covering, and that is called as epimysium. Okay. Next, from there, if you take one bundle where the muscle fibers are there, that is also covered by a connective tissue covering, that is called as perimysium. From there, take one individual muscle fiber, that is also covered by a connective tissue covering, and that is called as endomysium. Is it clear why this connective tissue covering is present? So that along with the connective tissue covering, blood supply and nerve supply can reach the muscle fiber. Okay, can you see the blood vessels and nerves? Ye dekho. Can you see? Okay, so that the blood vessels and nerves can reach the muscle fiber, that's why the connective tissue covering is given. Okay. So that is all about the skeletal muscle. Next is the cardiac muscle. So what do you know about the cardiac muscle? The cardiac muscle fibers? Yes? Now what about the fibers? How they are oriented? Yes, very good. As you can see in the diagram. Okay, so here also striations are present. The cardiac muscles which are there, striations are present, but the striations are not that prominent as compared to the skeletal muscle, but still striations are there. Can you see the striations? Striations rehta hai. Don't think that in cardiac muscle striations are not there. Okay, it is there. Next point is, if you take the fibers, so those fibers, they branch anastomose with each other. Individual fibers, they will branch, they will anastomose with each other. Next, can you see the position of the nucleus? Can you see the position of the nucleus? One nucleus is there, that also present at the center of the cell. Okay, one nucleus. And along with that, you can also see perinuclear halo. Perinuclear halo. Around the nucleus, a zone is there which is brightly stained under H&E, that is called as perinuclear halo, where different organelles are present in that particular area. Okay, so cardiac muscles. Okay, another important point which I have missed, sorry. In the muscle fiber, suppose one muscle fiber is there, another muscle fiber, they are connected to each other by intercalated disc. That is the most important striking feature for the cardiac muscle. So whenever you are writing in the exam or in practical viva, please mention that. Okay, this is connected to each other by intercalated disc. Why? Any idea why intercalated disc is present and what is the function of the intercalated disc? Yes, communication only. Actually, see, force of contraction from one muscle fiber can go through another muscle fiber through that intercalated disc. Okay, that's why the intercalated discs are present in the cardiac muscle cells. Now, I don't know you have seen the intercalated disc or not. So there are two components of the intercalated disc. Transverse component is there and longitudinal component is there. Okay. So intercalated disc are the complex adhering structure connect the single cardiac myocyte to an electrochemical syncytium. Now they are responsible for force transmission during the muscle contraction. Okay, so force of contraction is transmitted from one cell to another with the help of this intercalated disc. Now it is having three parts, that is the fascia adherens, macula adherent and gap junction. This is also called as desmosome. Okay, this is also called as desmosome. So cellular junction already you have studied in physiology. I know that. Same thing is present over here, but in a systematic way. So intercalated disc shows two components, as I said, a transverse component is there and lateral component is there. Okay, now the transverse component is perpendicular to the myofibril. If you take individual myofibril, it is perpendicular, the transverse component, and lateral component is parallel to the myofibril. Okay. Now, if you see the transverse component, it is in the form of staircase. Okay, CD jo hota hai, in the form of staircase. This particular transverse component is arranged. Now transverse component is made up of fascia adherents and desmosome, whereas lateral component shows the gap junctions. Okay. Now they allow action potential to spread between the cardiac cells by permitting the passage of ions between the cell. Okay, so action potential can spread from one cardiac myocyte to other cardiac myocyte with the help of this intercalated disc. Now here you can see the diagram. So this is how the appearance of the cardiac myocyte takes place. Okay, so you can see this is the fiber, you can see the striations. Can you see the striations? After that, can you see the nucleus, centrally placed nucleus? Then you can see the branching of the fibers takes place. And then here you can see the intercalated disc. See, these are the intercalated disc. Okay. Now, if you magnify the intercalated disc, generally not under normal microscope, for that electron microscope is required. At that time, you will see this structure. Okay. So you can see the transverse component and you can see the longitudinal component. Okay, so fascia adherents, desmosomes and gap junction, these are present. Okay, just now I have shown it. Okay, this thing you have not studied in physiology? Yes, I know you have studied in physiology. So go through it. I am not discussing in the class because we have to study the trachea also. Okay, now the striations, as I said, striations are also present. So the myofibrils which are there, okay, striations is there, and the striations are not that prominent as compared to the skeletal muscle. Now cardiac muscle fiber accommodates more amount of sarcoplasm and mitochondria. Okay, now this light band, dark bands, whatever it is present, it is same as compared to the skeletal muscle fibers. All those things are present. Now T-tubules are also there in the sarcoplasmic reticulum. Now do you know what are these T-tubules? Anyone? Have you heard about the T-tubules or transverse tubules? क्या होता है? Yeah, invagination into the from the sarcolemma, okay, into the muscle fiber. Why it is important? See, smooth sarcoplasmic reticulum of the cardiac muscle is not well organized. Okay, now these T-tubules are nothing but the extension of the sarcolemma. Okay, so the sarcolemma which is there, uska extension hota hai, that also transversely into the muscle fiber. That's why they are called as T-tubules. Now they penetrate the cardiac muscle at the level of Z line. Okay, that means the between one sarcomere only these T-tubules are present. Okay, so only one T-tubules per sarcomere in case of cardiac. T-tubules are present in skeletal muscle also, T-tubules are present in case of cardiac muscle also. Arrangements are different. Please note that. Here I am discussing about the cardiac muscle, not about the skeletal muscle. Now, along with that, the cardiac transverse T-tubules are highly branched invagination of the cardiac myocytes, okay, that are rich in ion channels for excitation, contraction, maintenance of the resting membrane potential, action potential, regulation and signaling transduction. These are the various functions which are taking place with the help of the T-tubules or transverse tubules. Okay. Now these T-tubules, whatever it is present in the cardiac cells or cardiac myocytes, they are forming a structure and that is called as dyad. That is called a dyad. In case of skeletal muscle, it is called as striated. Okay. So each T-tubule in the cardiac myocyte surrounding by small cistern, that is the dilatation of the sarcoplasmic reticulum, is forming a structure that is called as dyad. Okay. And thus small cistern and a T-tubule forming a structure that is called dyad at the level of Z line. Here you can see the organization of the sarcoplasmic reticulum, T-tubules and terminal cistern. Okay, so here you can see this is terminal cistern, this is terminal cistern, and here you can see this is the T-tubule. This is one T-tubule, this is one T-tubule. Okay, so these T-tubule are generally present only in the Z line in case of cardiac muscle. Okay, so this T-tubule and the cistern, terminal cistern which is present, they will produce a structure and that is called as dyad. Okay. Now this diagram is important, please note it because this you have to draw in a practical notebook. In practical notebook, I don't know whether you have drawn or not. If not, you have to draw this diagram. Okay, so this is the diagram of which muscle? Here this is the diagram showing the cardiac muscle. Okay, what you can see in this diagram? First you can see the branching pattern. Okay, you can see the branching pattern. Next, what you can see? Can you see the nucleus? Okay, centrally placed nucleus. After that, you can see the perinuclear halo also. ये देखो, perinuclear halo is seen. Next, can you see the intercalated disc? Okay, so these are the things which you have to identify. Okay, under the microscope. So this under 10x, this is 40x, and this is 100x. Okay. This is the actual appearance. Previous one, whatever I have shown you, that is a schematic diagram which you have to draw, and this you will see under microscope. Huh? This you don't have to draw. This is for understanding. This one you have to draw it. ये draw करना है. This you have to draw. Okay, and this for your understanding. That how the cardiac muscle fibers appears under microscope. Okay, muscle. So why it is called a smooth muscle? Any idea? Because yes, non-striated. Actually, see, striations are present, but it is very less prominent. That's why generally it is not seen under normal microscope. That's why it is called as striated. Next, what is the shape of the muscle fiber? Spindle shaped. Very good. Okay, it is spindle shaped. What are the location? As you can see, gastrointestinal tract, blood vessels, respiratory tract, urinary tract, erector pili, muscle, etc. Okay, these are the areas where these smooth muscles are present. Structure consists of elongated spindle shaped cell, and the size varies according to the location. Okay, now for example, in the smooth muscle cells of the blood vessel, it is 15 micrometer, wherever in case of uterus during pregnancy, the size increases. Okay, two sections are there. Whenever you will see the slide under microscope, two sections are visible. What are they? LS and TS. So the spindle shaped structure can be seen under LS only. In TS you will see circle. ठीक है, circular दिखाई देगा. But under LS the spindle shaped fibers can be seen. Okay. Next, here T tubules are not there. Okay. This is one important point. Centrally placed oval nucleus, spindle shape. Now, can anyone tell me, if T tubules are not there, then what is present? Instead of T tubules, क्या रह सकता है? What could be there? It is called as caveolae. Caveolae. Instead of T tubules, caveolae are present. Okay. See, this is the caveolae. So this is how the arrangement of the smooth muscle fiber takes place. So what is this shape? This is spindle shape. Okay. Now here you can see these are the invagination of the plasma membrane, and these are called as caveolae. Okay. Where in case of skeletal muscle you can see the T tubules are there. Can you see the T tubules? Okay. So these T tubules are absent in case of smooth muscle. Instead of that, caveolae are present. Okay. Same function, just the appearance is different. Yeah. So this is the diagram which you have to draw. Okay, this is the diagram which you have to draw. Now here, can you see which section is this? Longitudinal section, and this is transverse section. So transverse section का appearance कैसे होता है? Concentric manner होता है. And in case of LS, what is the appearance? Spindle shape. Can you see the spindle shaped fibers? Okay. So this also you have to identify under microscope. Okay. This is for drawing. You have to draw this diagram. So again you can see in different magnification the cell slides are shown. Okay, where both the sections are present. Transverse sections are also there. ये देखो नीचे ये, this is the transverse section and this is the longitudinal section. Now, though I have shown you the slide of this smooth muscle, okay, in the upcoming lectures we will be discussing about the GIT, structures of the GIT. For example, stomach is there, intestine is there, small intestine, large intestine. Okay? At that time I will not discuss about the smooth muscle. Directly I will tell that, see, in this particular part smooth muscles are present. Okay, at that time while looking at this you have to identify this type of appearance will be there. That also the muscle fibers are arranged in different ways. For example, some circular fashion is there, some fibers are arranged in longitudinal fashion. Okay, so different arrangements of the muscle fibers will be there. That's why repeatedly I am showing you this diagram. Okay, so please see the diagram and please remember that this is how the appearance is taking place, or else you will not understand the muscle fibers which are present in stomach, small intestine, large intestine. Okay. Okay, so that is all about the smooth muscle. Now this is the gist of the story. If you want to revise entire muscle chapter within 5 minutes, this is how you have to revise. Okay, everything is given. All the points, whatever we have discussed from the beginning. Okay, see the nature of the fibers, shape of the fibers, striation, then location, T tubules, then sarcoplasmic reticulums, specific structural features, connective tissue organization, location, key function, innervation, contraction, everything is given. Okay, this you don't have to write in practical notebook. Okay, don't write this one in practical notebook. So that is all about the smooth muscle. Actually it was a revision class. I know you have studied few things and few things you have studied in physiology. Okay, so I have combined it together and we have done a revision. Next is the trachea. Okay, so before going to the trachea, there are few important points which you have to know about the respiratory system. Okay, I hope you know what is respiration. You know what is respiration, right? And then you know that two types of respiration are there: external respiration, internal respiration. You have studied this. Next, the respiratory tract is divided into two parts. That is the upper respiratory tract and lower respiratory tract. Can anyone tell me what is present in the upper respiratory tract? Yes, pharynx, larynx, above the vocal cord. Okay, so above the vocal cord is the upper respiratory tract. Below the vocal cord, the lower respiratory tract starts. Okay, so in lower respiratory tracts, larynx, below the vocal cord, then trachea, bronchi, bronchioles, and then lung. Okay, these are the parts in the lower respiratory tract. Okay, so upper respiratory tract we will not discuss now. That will be discussed during head and neck portion after your this midterm. Okay, second term we will start with the head and neck at that time. Okay, so now we will discuss which part of the respiratory tract. Lower part, lower respiratory tract, that also from the trachea. Now here you can see the upper respiratory tract and you can see the lower respiratory tract. Okay, so we'll start from the lower respiratory tract, that also from the trachea. Now have you heard about this tracheobronchial tree? Anyone? Tracheobronchial tree क्या होता है? What is this tracheobronchial tree? So here also two parts are there. This is the conducting part and this is the respiratory part. Okay, this is very important. Why? Because if you are going below, that means from the trachea, if you are going towards the lung, at that time the structural changes are also taking place. Okay, in this pathway structural changes are also taking place. Along with that, histological changes are also taking place, starting from the lining epithelia, cartilage layer, muscle layer, then lymphoid tissue. Okay, everything is changing. So you have to follow that change. Then only you can understand the entire histology of respiratory tract, lower respiratory tract. Okay. So first you can see the trachea is dividing into two principal bronchi, that is right and left principal bronchus. Further you can see the two principal bronchi is divided into secondary or lobar bronchi. So this lobar bronchi, 3 in case of left lung, 3 in case of right lung, 2 in case of left lung. Why? Yeah, 3 lobes are there in the right lung and 2 lobes are there in the left lung. That's why the division is accordingly, secondary or the lobar bronchi. This for the individual lobes of the lung. So 3 lobes are there in the right lung, 2 lobes are there in the left lung. That's why the division is like this. Next, from the lobar bronchi, you can see the tertiary or segmental bronchi arises. Okay, which is 10 for both the side, right side also, left side also, and that will supply the each bronchopulmonary segment. Till now you have not studied, right? So there are 10 bronchopulmonary segment in each lung. Right side में भी 10 रहता है, left side में भी 10 रहता है. That's why here you can see 10 on each side. They are also called as tertiary or segmental bronchi, which will supply the bronchopulmonary segment. Below that, further they will divide into numerous bronchioles, and then again they will divide into terminal bronchiole, then respiratory bronchiole, then alveolar duct, after that alveolar sac, and then alveoli. Okay, so like this it will reach up to the alveoli. So this particular part is important. Today we will be discussing up to the trachea, and in next week we will discuss everything. Okay, so as you are going downwards, how the structural changes are taking place. Even the diameter is also changing. That means the passageway or the pathway, the diameter is also changing. Okay, so for that we have to follow all those things. Now what do you know about the trachea? From where to where it is extending? Anyone? Trachea is nothing but the continuation of larynx. Trachea is continuation of larynx. Esophagus is the continuation of pharynx. Trachea and esophagus, what is the relation? Trachea is situated anteriorly. Esophagus is situated posteriorly. Okay, don't forget, this is very important. Okay? Now, trachea is continuation of the larynx from the cricoid cartilage, lower border of the cricoid cartilage, that also from 6th cervical vertebra. Okay, then it is extending downwards, more or less it is present in the midline of the body. At the border of T4, lower border of T4 vertebra or 4th thoracic vertebra, it bifurcates into two principal bronchus. Okay, right and left principal bronchi. As you can see, and what is the length of the trachea? Approximately 10 to 15 centimeter. And the diameter is 2 centimeter external, internal diameter is 1.5 centimeter. External, yeah. Now the upper end of the trachea lies at the lower border of cricoid cartilage, as I said, at the level of 6th cervical vertebra, and in cadaver the bifurcation is lower border of 4th thoracic vertebra, corresponding in front of the sternal angle. Okay, that is the bifurcation. Now in case of cadaver, the bifurcated lower end lies at the lower border of the 4th thoracic vertebra. Okay, that is in case of cadaver. In case of living being, it goes below the T4, it goes up to T6. Okay, in cadaver it is T4, but in case of living being, it goes below T4 up to T6. Okay. Next, you know that the trachea, it is a fibroelastic tube. Okay, fibroelastic tube, and it is supported by cartilaginous skeleton formed by C-shaped ring of hyaline cartilage. 16 to 20 hyaline ring cartilaginous rings are there. Okay, and the posterior aspect of that ring is filled by a smooth muscle that is called as trachealis muscle. तो ऐसे C shape रहता है, ठीक है? तो anteriorly and posteriorly the cartilage is present. Anteriorly and laterally. Posteriorly gap is there, and that gap is filled by trachealis muscle, which is a smooth muscle. So why I am telling you this? Because in the slide you have to identify the cartilage also, which we have studied earlier, and you have to identify the smooth muscle, which we have seen today. Next, the lumen, which is lined by respiratory epithelium. What is the lining epithelium? Pseudostratified ciliated columnar epithelium, or also called as respiratory epithelium. Okay. Next, so trachea is a tubular structure. Trachea is a tubular structure. You have seen that. Now, if a tubular structure, if I take a TS, what will be the shape? Circular. As you can see, it is circular. Okay. So here you have to identify which part is anterior, which part is posterior. तो ये वाला जो part है ये anterior होगा, ये posterior होगा. How will identify? How will differentiate? Yes, because of the trachealis muscle. Trachealis muscle कहाँ पे रहता है? Posteriorly रहता है. As you can see, the trachealis muscle is present over here. Okay, so this will be the posterior aspect. This will be the anterior aspect. Okay, and on the lateral aspect also the cartilage is present. Next, you have taken the TS of the trachea. You have prepared the slide. Okay, so always keep that in mind whenever you are taking TS. At that time, various layers are there. in histology, whenever you are preparing the slide, various layers will be there. It depends, sometimes 4, sometimes 5. So here how many layers are there? 4 layers are there. What are those layers? First you can see the mucosa is there, and the layers you have to mention specifically. That means from inner layer to outermost layer, or outer layer most layer to the inner layer. You can't mix it during the time of exam. If you are mixing, that means you are wrong. Okay? Either you should go from innermost layer to outermost layer, or outermost layer to innermost layer. Okay, this serial should be maintained. Now you can see the innermost layer. First is the mucosal layer, then is the submucosa, then cartilage and muscle layer together. Okay, muscle is present on the posterior aspect, followed by the adventitial layer. So now you have to know what are the things present in these particular layers. Okay, we are in systemic histology now. General histology is over. Now we are in systemic histology. Whatever we have studied will be applied over here. So first is the mucosa. तो mucosa में क्या रहता है? Lining epithelium. Okay, so the trachea is lined by pseudostratified ciliated columnar epithelium. Okay, also called as respiratory epithelium. And you have studied, if any epithelium is there, then basement membrane will also be there. As simple as that. So see here, resting on the basement membrane. Along with that, along with the lining epithelium, another type of cells are there, and these are called as goblet cells. Numerous goblet cells are present. Okay. Now, how will identify between the lining epithelium and goblet cells? Any idea? How will identify? When you will see the slide, lining epithelium, I know you have studied about pseudostratified. You know the features of pseudostratified. If no, go and study. Now you have to apply over here. Okay? So pseudostratified epithelium, once you have identified, you will see that in between the slide there are some spaces, empty spaces. कुछ नहीं है, white color का empty spaces है. Why? Because mucus was present, and during the slide preparation the mucus is washed away. So if it is washed away, it can't take the stain. That means it will appear empty. This is how you have to identify the goblet cells. Goblet cells directly दिखाई नहीं देता, you can't see. Okay. Next, the cilia is present. Each cell supports approximately 220 to 270 cilia on its surface. Okay. Then mucus, which is secreted by the goblet cell, will entrap the dust and other foreign particles. Now these entrapped particles are thrown away by the ciliary beads towards the upper part of the trachea and the larynx, and that is ejected out by cough reflex. Now the question is, why cilia is present? जो respiratory epithelium है उसमें cilia क्यों रहता है? Why? See, the cilia which are present on the respiratory tract, mainly the respiratory epithelium, those cilia, they are beating. The beating of the cilia is producing a wave, and it is in one direction towards the larynx. Okay, any foreign material goes inside, it will be first entrapped by the mucus secreting cells. Then because of the beating of the cilia in particular direction, it is towards the larynx. After that, the person can cough and spit it out. That's why the cilia are present, and ciliary movement is very important. Okay, in chronic smokers, this beating is affected. Okay, person with smoking regularly for a longer period of time, this beating is affected. Okay, now please note that here I have mentioned that two types of cells are there. That is the lining epithelium, along with that goblet cells are there. Okay. Next, another types of cells are also there. Please note it down. These are called as brush cells. Brush cells and enteroendocrine cells. Two types of cells are also present. Okay, these are called as brush cells. Okay, brush cells and enteroendocrine cells. जो brush cells होता है, on the surface of the brush cells, this blunt microvilli are present. Okay, they are chemosensory. When a irritating material goes inside the respiratory system, this particular cell will sense it and will produce cough reflex so that it can be thrown out of the respiratory tract. That is the function of the brush cells. Okay. Along with that, another type of cells are there. As I said, these are called as enteroendocrine cells. Okay, उसका function क्या है? They secretes a compound that is called as catecholamine. Catecholamine. Okay, they are nothing but bronchodilators. Okay, when stressful inspirations are there, at that time it will dilate the respiratory passage, so that more amount of air can go in. Okay, so these are also the cells which are present in the mucosal lining. So you have to mention it. Okay, what are the two cells which I have mentioned? Brush cells and enteroendocrine cells. Along with that, the functions also. Okay, another type of cells are there, that is called the basal cells. These are having stem cell functions. Okay, basal cells are also present having stem cell function. Okay, next, the basement membrane is there. As you know, if there is any lining epithelium, it needs basement membrane. Okay, so for that the basement membrane is also there. Now the basement membrane present a glassy homogeneous eosinophilic layer. Below the basement membrane, a loose connective tissue layer is present, that is called as lamina propria. In the mucosal layer itself, just below the basement membrane, a loose connective tissue layer is present, that is called as lamina propria. Okay? In that layer, the speciality is BALT is present, BALT. Now what is this BALT or BALT? Bronchus associated lymphoid tissue. Bronchus associated lymphoid tissue. Okay? That is present in that particular layer. That is the speciality of this layer. So it shows many diffuse lymphocytes and lymphatic follicles in the form of bronchus associated lymphoid tissue. Okay. In case of lamina propria, the elastic fibers are also present. Okay. Now these elastic fibers which are present, generally it cannot be seen under H&E stain. For that special stains are required, that is called as oracin stain. Previously we have discussed elastic fiber you can't see under normal H&E stain, very difficult. For that special stains are there, that is called as oracin stain. Okay. Next, submucosa is there. So submucosa is situated below the lamina propria, where you can see the mucus and serous glands. Okay, mucus and serous glands. Now can anyone tell me the difference between the mucus and serous gland based on their secretion? प्लीज कीप दैट इन माइंड. जो सीरस ग्लैंड होता है उसका जो सिक्रेशन है दैट इज वाटरी इन नेचर. ओके, और जो म्यूकस ग्लैंड होता है दैट इज थिक विस्कस इन नेचर. तो यहाँ पे व्हाट इज द फंक्शन ऑफ सीरस ग्लैंड? द सिक्रेशन विल कीप द एपिथेलियम मॉइस्ट. ओके, or else the epithelium can die also. Next, the mucus secretion, whatever it is there, that will entrap the dust particle or any foreign material which is going inside the respiratory tract. That's why in the submucosa you will see that round structures are there. These are nothing but the mucus and serous glands. Okay. Along with that, it also contains blood vessels and lymphatics. Okay, submucosa mein blood vessels and lymphatics are also there. Next layer is the tracheal and cartilage layer, where 16 to 20 cartilages are there. So hyaline cartilage, all the features of the hyaline cartilage are present. Perichondrium, cell nest, lacuna, okay, everything. Ground glass opacity, that is the homogeneous appearance. Okay, all those things are there. Next, it consists of chondrocytes. Okay. Now on the posterior aspect, as I said, a smooth muscle layer is there, that is called as trachealis muscle. Okay, which is a smooth muscle. Now can anyone tell me why this trachealis muscle is present? Why it is not completely made up of cartilage? जो ट्रेकिया होता है उसमें जो कार्टिलेज रहता है सी शेपेड रहता है. Why? Okay, instead of C shaped, why it is not complete circular ring? Yes. No, you are very near to the answer. Okay, so see, I have told you that the esophagus, the trachea and esophagus, how is the relation? Anteriorly क्या रहता है? Trachea रहता है. Posteriorly क्या रहता है? Esophagus. If you see the lumen of the esophagus, it is always closed. हमेशा बंद रहता है. Whenever you are swallowing something, at that time it will open, and then again it will close. Okay. Now whenever it is opening, at that time you need some space. Though it is situated just posterior to the trachea, so that space is provided by the trachealis muscle, so that it can expand. Then only you can swallow things easily. Okay. And lastly, the adventitia, fibroelastic connective tissue containing large blood vessels and nerves. Okay, that is present in the adventitial layer. So whatever we have discussed, this is the diagram. Okay. Now can anyone tell me which is the innermost layer in this diagram? This side or that side? Which one? Which side? This side or that side? This side, this side. This is the innermost layer and that is the outermost layer. Okay. So how many layers are there in the trachea? 4 layers are there. What is the first? Mucosa. In mucosa, mainly what you have to see? The lining epithelium. So can you see the lining epithelium? So what is this? This is the lining epithelium. In lining epithelium, can you see some small empty spaces? These are nothing but the goblet cells. Okay. Next, this particular part is the lamina propria. तो जहाँ पे epithelial cell खत्म होता है, in that particular part you will be getting the basement membrane. Just deep to the basement membrane, the loose connective tissue which is present, that is the lamina propria. Okay. In lamina propria, what is the speciality? Bult is present. Bronchus associated lymphoid tissue. These are nothing but the aggregation of lymphoid follicles. Okay. So that is this lamina propria. Okay. Next, if you go more deeper, then this is the submucosa. In submucosa, can you see the glands? ये देखो, this is how the gland appear. Okay, this is how the glands appear. Okay. Further, if you go more deep, more posteriorly or more outerwards, then which layer is this? Which layer is this? Cartilage layer. This is the cartilage layer. This is how the cartilage appears. You can see the perichondrium. You can see the cell nest, everything. Okay. Beyond that, if you go, then this is the adventitial layer. Okay, adventitial layer may, as I said, mainly blood vessel nerves are there. Some adipose tissues may also be present. Okay. Now here you can also see the trachealis muscle. So can you see the spindle shaped arrangement? Okay. So this is how you have to identify the slide under microscope. Okay. So this you have to draw. This you have to draw in your practical notebook. ये draw करना है, ठीक है? And this is for your understanding. So by looking at this slide, okay, you have to identify under the microscope. So here, can you see the lining epithelium? Then you can see the basement membrane. You can see the lamina propria. Then you can see the submucosa. Which layer is this? Cartilage layer. And then you can see the adventitia. Okay. So I hope you have seen this diagram. Now we'll identify. Okay.